This is a video for topic 2.3 in the IB Biology syllabus. Here are the understandings, applications and skills for this topic. As you can see, it is all about sugars, carbohydrates and fatty acids or lipids. There we are. So starting off with what a monosaccharide is just a single sugar subunit. These can then join together with a condensation reaction, shown here, where two H's and one O is taken out. So a water is taken out and a bond, the glycosidic linkage with the O in the middle, creates a disaccharide. So on top of disaccharides, you can also have polysaccharides, which would just mean this chain just keeps extending down this way, with a water coming out every time the bond is formed. So monosaccharides are mainly used for energy sources, disaccharides for transport, and polysaccharides for storage form. And that's just because of how easy they are to access. So there's some examples of each type. Starches are cellulose and glycogen are all examples of polysaccharides. So these ones are found in plants, glycogen found in animals. There's two types of glucose, so beta glucose and alpha glucose, and that's which ones those ones are made of. They're bonded in different ways, where they're branched, so different branches coming off, or just in one straight chain. And their different structures contributes to their very different functions. So here cellulose is a structural component, starch is energy storage, and glycogen is also an energy storage, but found in animals. So fatty acids are long hydrocarbon chains that are found in certain types of lipids. Fatty acids may differ in the length of hydrocarbon chains, so they're usually about 4 to 24, but they can be bigger. And they also vary in the number of double bonds. So here we have types of fatty acids. So saturated would be these lines, and the corners represent carbon atoms, and it's implied that there's two hydrogens bonded to each carbon atom along this chain. Monounsaturated means there's one double bond, meaning there's less hydrogen atoms bonded to this chain, and polyunsaturated means even fewer hydrogens bonded to this chain and more double bonds. There's different types of double bonds in a fatty acid chain. As you can see here, trans is where the hydrogens are bonded to opposite sides of the double bond, and cis is where they're bonded to the same side of the double bond. Trans fatty acids do not occur commonly in nature and are produced by industrial processes called hydrogenation, and they're generally linear in structure. Triglycerides are the largest class of lipids. Animals tend to store triglycerides as fat, while plants tend to store triglycerides as oils. They're formed when condensation reactions again occur between one glycerol and three fatty acids. The hydroxyl groups of the glycerol combine with the carboxyl groups of the fatty acid to form an ester linkage. This condensation reaction results in the formation of 3 molecules of water. So here's this reaction here: the glycerol, 3 fatty acids, and 3 condensation reactions, leading to 3 waters as well. Here's the different types of fats and how healthy they are. There's huge amounts of research that goes into this, but it's still rather subjective as to how bad fats are for you, and there's been a lot of debate over time. So here you can see cis fats are good, trans fats are bad, saturated fats are bad to a certain extent. So the thing with cis fats is that they're bent, that the two hydrogens on the same side cause a bend, whereas these are straight, and that means that they can build up inside of arteries more easily by piling up against the wall, whereas the bent ones are less likely to get stuck, causing blood clots. So different examples of foods with good fats, raising good cholesterol levels. Saturated and trans fats raises bad cholesterol levels, which can lead to heart disease and other problems. So the differences between carbohydrates and lipids, shown here. So short-term energy storage, long-term energy storage, digestion, much easier to access carbohydrates, less easy to access lipids, much less energy in carbohydrates compared to lipids, and then solubility in water. Finally, body mass index, calculated by mass in kilograms over a person's height in meters squared. So that provides a measure of relative mass based on the weight and height of the individual, and it's a commonly used screening tool to identify potential weight problems in sedentary adults. So the ranges for numbers range from underweight to obese according to predetermined values, but there are, however, flaws with the system, as BMI values are not a valid indicator for pregnant women or professional athletes or anyone with a significantly atypical body type. They should not be used as a diagnostic tool, and these days doctors have come up with much better ways of deciding how unhealthy a person is. But there is topic 2.3.